So that's the goal for today. First, let's start with a bit of review. So what were the top points from last class? What do you remember most about talking about last class? So karyotypes, what's the other karyo word? Karyotypes and karyograms, and we talked about the distinctions between the two. Does anybody remember? Which one's the one that's a drawing or a picture? That's the karyogram, right? It's like Instagram. It has to do with pictures. The karyotype has to do with typing, writing, right? The written description of the karyogram. We also talked a little bit about using fluorescence in situ hybridization experiments to address hypotheses about chromosomes, chromosomal relationships, and so forth. We're going to use a little bit more of this technique later in class today. Is there anything else from last class? Karyotypes, karyograms, fluorescence in situ hybridizations. There's one other technique we talked about. We didn't talk about it very much. So in addition to fish, cytogeneticists have one other tool that they use more frequently, actually, to understand something about what chromosomes look like. So we talked about DAPI staining. DAPI is a chemical that scientists apply to DNA to actually be able to see it under a microscope. And we talked about other chemicals, not specifics, but other chemicals that scientists also apply to chromosomes, and that's to elicit what? A banding pattern. So we talked about using DAPI, a fluorescent chemical that we can use to see DNA. We also talked about how other chemicals, with no detail in particular, let us perform chromosome banding. And what's the main reason we would want to look at banded chromosomes as opposed to unbanded? What do the bands do for us? Right, so what's true about chromosome bands? If you look at human chromosomes and they're banded. So the answer up front was they help you identify the chromosome. So what's true about our 23 different chromosomes and their banding patterns? Right, so for homologous chromosomes, that would be the version of chromosome 1 you got from your dad and the version of chromosome you got from your mom. Despite the fact that they are going to have different sequences because those are chromosomes that came from two different people, they will have the same banding pattern. And that's how we can use chromosome banding to make karyograms. You can see the banding patterns. You pair up like chromosomes, like we've been doing since kindergarten. Not with chromosomes, but with other things. And that's how we make a karyogram. So you can use the banding patterns to identify what chromosome it is you're looking at. So one of the exercises from last class was, here's the Muntjac metaphase chromosome spread, here, for example. So draw me a karyogram. So if you had digital image editing software and you could actually cut out and spin the pictures of the chromosomes, draw me what you think you would make as a karyogram. So these are a selection chosen at random of your responses. So let's number them for ease of quick discussion. 1, 2, 3, and 4. For you personally, which one do you like the best?
3.
So I hear 3. Anybody else? 3. I hear a lot of 3s. Good job, student who submitted number 3. Anybody else? Number 2. Okay, so what's different about 4? I like 4. I like the use of color. It helps me see, right? And this is not something you would see in a normal karyogram, but it helps you figure out, which chromosome it is they're trying to represent when they draw a cartoon of it. That I like. So why isn't anybody saying 1? There are too many chromosomes. So I think I know, I'm guessing I know what picture 1 shows. It's a karyogram. Well, It's a type of a karyogram, as all of these are. There's a third term that I want to introduce you to. The reason that we're talking about this is both to get feedback. So this is a step where all of you submitted drawings, or most of you anyway, of karyograms. I'm going to introduce a new vocabulary term, and the other point that I want to make now is we're going to do this a lot this semester. When you draw something or annotate something and send it to me, I'll show a small sample of results, the answers up here. Because I don't give every single one of you individual feedback on these exercises because of the sheer number of you, and there's only one of me, this is your great chance for you to see, so I hope you're actively thinking about what did your drawing look like compared to these? So I can't give you individual feedback on every one of your submissions, but you can at least compare what you drew to what's up here to try to get feedback on your understanding of this concept. So my guess is picture 1, 23 pairs. So what's this a karyogram of? It looks like a human karyogram, which is totally fine. So a student that submitted this one demonstrates to me that they understand what a karyogram is. It doesn't really matter to me that the point of drawing a muntjac karyogram was missed. It's okay. Right? Clearly understand what's going on. So the vocabulary term. An ideogram. Sometimes you see it spelled I-D-I, sometimes you see it spelled I-D-E, doesn't matter. So an ideogram is what we've actually done here. An ideogram is the term we use for a drawn or a cartoon version of a karyogram. Right? I'm never testing on facts. There's not going to be vocabulary on the test. I just want you to have heard this term once, that really we reserve the term karyogram for photographs of chromosomes that have been rearranged. Whenever we do representations of chromosomes, then we call it something else, an ideogram. Any concerns or questions up till now? So that's all review from last class. Catching everybody back up to speed. So for today, the goal was to have read the book and also to have watched a video on a few different types of chromosomal rearrangements. This all, of course, pertains to that first question: how do you go from the structure of genes on a chromosome in a horse to a chromosome in a donkey? Really briefly, the first step here is to work together to come up with a group understanding of what is it that each one of the 4 different types of chromosome rearrangements actually looks like. What's the process? What's the result? So we'll start with the easy one. At least, I think it's the easiest one. So say you have 2 genes on a chromosome. Well, let's do 3 genes just for fun. So chromosome, whatever this is, has 3 genes, gene 1, gene 2, gene 3. If there's a duplication of gene 2, what am I going to draw on the right-hand side? So we duplicate gene number 2. What's the result? When we talk about cytogenetics and a duplication of a part of a chromosome. So what does it mean? What has the cell done if we talk about a duplication of part of the chromosome? What happened inside the nucleus? Right, it's duplicate, you've got 2 of them. So you've got 2 gene 2s. So where do you want me to put the second copy of gene 2?
Next to the first gene 2.
So here, next to the original gene 2, that's fine. Could it go anywhere else? Could I draw it up there? It doesn't matter in sum where the gene lands, this extra copy of chromosome 2, we just know that it's somewhere on this chromosome. Duplication doesn't even have to insert itself onto the same chromosome. So when we talk about any sort of chromosomal duplication, that duplicate copy of whatever part of the chromosome we're talking about could land anywhere in the genome, on any chromosome, that is. So what has to have happened inside the cell to go from the chromosome on the left to the chromosome on the right? What physical processes had to have happened? to the DNA. How do you make a copy of DNA? What process does that involve? One word. Replication. So somehow a cell screwed up and made an extra copy of a part of a chromosome when it was replicating its DNA. So that's the duplication part. So this happens all the time. So don't think this is weird or crazy or rare. It's pretty rare, but it happens. Cells screw up and make extra copies of parts of chromosomes. So if you've got an extra bit of DNA floating around in the nucleus, that would look like this, potentially. There's an extra copy of part of this chromosome carrying gene 2. How do we go from that step to what's drawn on the right? What has to happen? How do we go from one copy of chromosome 2 in the middle of a chromosome to 2 copies of gene 2 in the middle of the chromosome? How does this bit of DNA get in there? What physically has to happen to the chromosome?
Does it have to split first?
It has to break. Right? How are you going to put a piece of DNA in the middle of a chromosome? That chromosome has to be broken to stick that extra piece of DNA in. So you might imagine in this case that there was a break right there. And that this piece of DNA then inserts itself where the chromosome broke. So that's, in its simplest form, a duplication, sometimes also called, it's a form of an insertion. A piece of DNA sticks itself into the middle of a chromosome. So conceptually simple, you got a chromosome, you got an extra piece of DNA. You stick the extra piece of DNA in the middle of the chromosome or on one end. It's deceptively simple, though. So what questions do you have? I see a lot of concerned faces. Any concerns or issues about this? Sir? So first, I'm going to interpret your questions. How do you get the chromosome to break? Don't blame me, man. I didn't break the chromosome. So in addition to the fact that the cell sometimes accidentally makes extra copies of DNA, chromosomes also break for no particular, well, they're for a particular reason, but not because it's caused by human action, I want to make clear. So, for example, those of us that suffer from lack of melanin in the skin, us white people, when we get bombarded with ultraviolet light, that ultraviolet light can do things to our DNA like break the DNA. Right? High energy photons can actually physically break a piece of double-stranded DNA. So if you happen to be a cell that's been hit by ultraviolet light and there happens to be extra DNA floating around at the same time, our cells have repair mechanisms that try to find 2 pieces of DNA ends and stick them back together. And if it happens to be that those cellular enzymes grab the wrong 2 ends of DNA and stick them together, that can be how that leads to an insertion. So all the cell's trying to do is repair itself. It recognizes that it's got broken bits of DNA floating around in its nucleus. It just sticks them back together. Because, of course, our cells don't have brains, so they don't know what they're doing, really. And that's one way that those chromosome breaks happen. So natural causes, many different types. Ultraviolet light's one of them. So I don't ever mean to imply that this is happening for a purpose. All of these processes are happening randomly, by chance, essentially. Which parts of chromosomes duplicate, that is, you get an extra copy of, random. Where the chromosome breaks, how many chromosomes break in any one cell at any one time, random. So this just happens. It's rare, but it happens. Yeah,
So the duplication is random?
the duplications are random. They don't happen very frequently, but they happen at a rate that's high enough to cause diseases in some cases. So you get rare people with rare diseases that involve duplications of certain genes. So you've got one extra copy of one gene. Maybe that causes a disease. And so the reason we can detect these really rare events is, I think I've mentioned this before, when humans get sick, they go talk to their doctors. So humans are actually the species where we have some of the best estimates of these sorts of chromosomal rearrangements, because when they cause disease, our physicians collect the data and tally them up, and we know the incidence of some of these types of rearrangements. Duplications of certain parts of the genome, deletions, translocations, the things we're talking about today. Other questions? Yeah.
Are there ever any changes that are intentional to the DNA? Like any rare cases where something selected that?
So you use the word selected. So you're talking about
Okay, within one organism.
So
not
within an
multiple
organism,
generations.
let me rephrase. Are you asking, do our cells ever intentionally rearrange our DNA?
Yes.
Yes, is the answer. See, I suspect nobody in here has taken immunology yet. Does anybody know anything about the immune system? Turns out that there are certain parts of our genome where our chromosomes do actually get rearranged, and part of that has to do with, so there's an instance of immunology. Our antibody genes physically rearrange themselves to produce the number of diverse immune molecules that we have for recognizing antigens. Right? So our body defends us by having a diverse number of antibodies that go out and look for all sorts of different shapes of objects in our blood and tissues that might represent threats. And the way that the genome handles that and produces that variety of genes to produce those antibodies is through breaking and rearranging parts of our genome. There's one example of an intentional time that this happens. So isn't that weird? Our genomes, follow me here, it's not too bad. Our genomes encode genes whose job it is to encode proteins that intentionally break up our genomes and rearrange them. It's sort of a suicidal kind of thing, except it's really strictly controlled genome rearrangement in the case of the immune system. There are other times when you don't want these processes to go haywire, right? You don't want to turn your DNA rearranging genes loose in your genome. They have to be really tightly regulated. We'll get into some of those examples later in the term. Okay, so with the concept of chromosomes breaking in mind, how does a deletion work? At its most basic form. We start with a chromosome that's got 3 genes, we wind up with a chromosome that's got 2 genes. What happened to gene B? So what physical process has to happen for that transition? Again, by chance, randomly, what happens to the chromosome on the left side? First thing.
It's broken.
Okay, so it gets broken, thank you. How many times does it have to break? Twice. Where does it have to break?
On both sides.
On either side of gene B, right. So somewhere in between gene A and B, and somewhere in between genes B and C, random breakages have to happen. So an intermediate state might be, here's a fragment of a chromosome with gene A, here's a fragment of the same chromosome with gene C, here's a fragment with gene B. So 2 breaks, that's an intermediate state. The cell repairs this by, unfortunately for this person, trying to repair the damage by sticking the ends of the chromosomes near genes A and C together and forgetting all about the part of the chromosome that contained gene B. Do you think this is good, bad, or doesn't matter to the cell?
It could be bad because now there's like a completely different rearrangement of the gene sequence.
Okay, so the gene sequences have rearranged.
But it depends on what the gene coding is for to make that decision, whether it's good or bad or neutral.
Yeah, there are a lot of potential problems here. So it's hard to say whether or not this is neutral or good or bad. To me personally, it doesn't seem like this is likely to be a good thing. Usually my perspective and a lot of geneticists' perspectives is the chromosomes are arranged the way they are for a reason. That doesn't mean they can't be improved on by change. That's evolution. But when you start breaking or rearranging chromosomes, sometimes bad things happen. We're going to look at one example in just a minute. So that's a deletion. Chromosomes break, parts of chromosomes don't get reassembled together after the chromosomes break. Okay. Same principles, inversion. If I say that genes, the chromosome part, the segment of chromosome that contains genes 2 and 3 inverts, what does that mean?
That it broke away and then it reversed and reinserted itself into it.
Okay, so there was breaking, you said reverse, inverse, reverse, flip is a reasonable word to use. So 2 breaks to the chromosome, in this case. That whole section of DNA just rotates. I mean, it's in the nucleus. It's in liquid. It just sort of floats there and it turns up. Well, I can't turn all the way upside down. It turns upside down, and then the cellular machinery comes and tries to fix the problem. It accidentally puts this part of the chromosome back in upside down relative to the original state. So these are all things that happen in cells all the time. Small ones, big ones. Big ones usually cause problems. Maybe small ones don't. So that's another process. All it involves is chromosomes breaking and chromosomes being reassembled in different orders. So here's the one that's a little bit the odd one out. Translocation. Does anybody know why this one differs from the 3 cases we've looked at already? So now we're involving multiple chromosomes. So before, all we've talked about so far is one chromosome changing. For a translocation, that means now we're involving more than one chromosome. So 2 chromosomes, 4 genes. What's a type of a translocation? We could get breakage and rearrangement so that gene 3 winds up on the same chromosome as genes 1 and 2. Using all the same processes. The only difference now is we're making chromosome breaks, but instead of the cell fixing those problems, so what happens is there's a break there in between genes 3 and 4, for example, and that fragment, the cell decides to stick on the end of a chromosome. Using exactly the same mechanisms and forces, random events that happen with all the first 3 types. Now I've just involved a second chromosome. In fact, it doesn't have to be end to end. You could also maybe envision the idea that there's also at the same time a break between genes 1 and 2. Gene 3 could be put in the middle. So you could wind up with gene 3 translocating to the middle of the other chromosome. That's fine. It's a translocation, moving trans across from one chromosome to another. Okay. Deletion, inversion, translocation, duplication. Now let's put it to practice. So here's an order of genes on one chromosome. So you're looking at a horse chromosome, that order of genes, A, B, C, D, E. In a donkey, you look at the positions of the genes on the chromosome, you see something totally different. So for the next 5 minutes or so, on your own or in groups, it doesn't matter, just try to figure out, how do you get from step 1 on the left to step 2 on the right? What sorts of rearrangements, how many, and if you really want to get into it, what order do they occur in? Okay. Go ahead, and we'll come back together in 4 or 5 minutes. And that's why I do want to break up the conversation now so that we can get some feedback from you and you can ask more questions if you want. So how did you start this process? What was the first thing you did? So you want to break some chromosomes. Yes. What's the most obvious change to you? There's no right answer between the initial and the final states. What do we tackle first? Sure. So I heard C and D aren't present over here on the right. So what do you want to do with C and D to get rid of them? Let's delete them. When do you want to do that first? Does it matter when we do the deletion? It might. So when we get to the end, we'll see. So first step, let's say we delete C and D. That involves breaking the chromosome between C and B and between D and E. So what sort of a chromosome does that leave us with? What's the order from top to bottom? A, B, E. Okay. So if that's your hypothesis, which is totally fine, you can have other answers, that's fine too, as long as we make it to the stage on the right. So what are we going to do now to get from the middle to the right side? Duplication of A and B. So you make an extra copy, the cell makes an extra copy of that fragment of the chromosome. Now what? So it's got to insert itself into the chromosome. So where do you want to break the chromosome? Between what genes? Between B and E, you want to make a break. And we're going to stick that little extra fragment, the extra copy of A and B in. What has to happen now? So this A and the B are going to go in the upside down orientation. So they're going to rotate, and then that's going to stick in there, and that takes us to the final state. So we've used, so far, a deletion, a duplication, coupled with an inversion. This is not the right answer. This is a right answer.
Do you think that it would be better if when you have it duplicated first, and then when C and D break off and do the complication, then A and B would come in? Just because like there's not as many breakages?
Yeah. So that's an excellent question. And this is why I'm going to be constantly harping on everybody to understand that there's not always one right answer. There are lots of ways to get from step 1 to step 2, from the start to the end. So the hypothesis here, the proposal is you duplicate A and B, but instead of sticking this copy in down here, as we showed it, what if it inverted and stuck itself on the end of the chromosome, upside down? Oops, not upside down. Does that get us anywhere closer to what's on the right? So the goal is to try to find the smallest number of changes that you need to go from the initial step to the final step. We want the fewest number of changes, the fewest number of chromosome breaks and deletions or duplications. Why? Why smallest number?
Less energy.
Less energy used, better chance of it happening. If every one of these steps, genome accidentally duplicating part of itself, chromosome randomly breaking, the more of those events that you require in your model, the less likely all of those are to have happened by chance. So the way we approach this usually is to try to come up with this path, the series of events, we're modeling what might have happened that require the least steps. So you might have come up with a totally different solution, which is absolutely fine, as long as it's got no more than, what, 3 chromosome breaks and 1 duplication. There are ways to do it with way more chromosome breaks. And if you came up with one that requires less steps, please let me know. Because if so, then I haven't shown you the most likely model for how this happened. Sir?
I might be thinking about it too much, but wouldn't A and B...
There's no way to think about this too much. What are you talking about?
Would A and B actually probably want to be inserted separately from each other? Because if it's going to be inverted, wouldn't that change the actual coding sequence of it?
So it depends. So the answers to all of those questions depend. Is it likely that A and B invert or not? May matter, may not. So it depends on real details of the situation, which I haven't provided. I haven't provided you enough details to know what sort of scenario would be more likely. And lest you be worried about how I might grade this on an exam when I give you this type of a question, I want you to know now that if you come up with a reasonable answer, you get the points. Because there's no right answer. So never go into one of my exams thinking every question there's going to be one answer and that's it. So it takes me longer to grade the exams because I have to look into the details of what you've proposed. There's not just an answer key and yep, that's the right answer. So I hope you'll be comfortable with that concept. It's difficult because that's not what most students have experienced prior. But as long as you come up with a reasonable explanation, that's fine. So if you invert A and B in the first step or you invert them in the last step, may not make a difference. So what I encourage all of you to do, final word on this subject, what I encourage everybody to do is to play around with the order of events that we drew up here, I drew up here. So for example, after class, try doing an inversion first and then a duplication, or try doing the deletion of C and D at the end rather than at the start. See if that matters. Does the order matter? Try it on your own.
And if it duplicated before it reattached to E and connected and then it just connected to the top, that's only 2 breaks.
Right.
So
That would be better.
Yeah.
The way that I've drawn it here in blue? Or are you envisioning something else? So if A and B duplicated, they flipped upside down and they attached on to the end there. Am I misunderstanding what you're proposing?
Maybe. I'll just...
Well, do please tell me. Not necessarily now, but if not, after class.
Okay.
Because I want to bring this back to everybody in case there is a simpler way to do this. And that goes for everybody. If you come up with a better way to do this, let me know. So the bulk of the work that we've done today is to address the top point, to understand, to propose a way to come up with a chromosome rearrangement or a series of chromosome rearrangements that takes us from the structure of one chromosome to the structure of a related chromosome. Yes?
Question about the other thing.
Please.
When it's duplicated and it's separate, can it flip on itself there and then go back in or no?
Yes. So when that extra piece of DNA, the A and B duplication, when it's floating around in the nucleus, it can insert itself in any order, A up or B up.
Okay.
Yeah, because it's just floating in the nucleus.
Yeah.
So anything is possible at that point, either orientation.
So if we go back and, because there's 2
And then 2 breaks.
One break in between
Both of them?
both, yeah. Okay, so in the interest of time, Or any other questions? There is a lot of conversation, so I want to make sure that we're clear here. Any other questions? So I'm going to tell you briefly one story about a translocation that actually happens, not something that I hypothetically made up. The Philadelphia chromosome in humans. We're going to skip over chromosome fission and fusion for today. We might come back to that next class. So a couple more points about chromosome nomenclature. Here's chromosomes. What are these, by the way? These are drawings of chromosomes. Good to remember from the start of class. It's an ideogram. It's an ideogram of banded chromosomes. So it's a drawing of what an actual chromosome would look like when it's banded. So these are ideograms of human chromosomes 9 and 22. What do we call this short arm of the chromosome? What's its abbreviation, or what do we call it? This is the P arm, and the long arm is the Q. So that explains what that Q is doing there, the 9q in this nomenclature of cytogenetics, chromosome 9 Q arm, long arm. Why P and why Q? P stands for petite, small. Q stands for one of two things. Either it comes after P, which is one answer that I've heard from cytogeneticists, or it stands for Q, like queue up, like form a line, long line, queue up for the movies. I know it's British, but still, work with me. So P arm, short arm, Q arm, long arm. Where's the centromere? It's in between the P and the Q arms, right? It's where there's this little constriction, narrowing drawn on the chromosome, or we see it when we look at an actual photograph of a chromosome. So if the centromere is off to the side, there is a P arm and a Q arm. The centromere makes a short arm and a long arm. What kind of a chromosome is this? What's the word for its shape or structure? If the centromere is in the middle, it's a metacentric. RIC, metacentric. Centromere in the middle. If the centromere is off to one side, it's either submetacentric or if the centromere is really towards one end of the chromosome, it's acrocentric. So if these aren't familiar to you, I suggest looking in the textbook just to read the definitions of these. If the centromere happens to be at the telomere, that is, there's a telomere and there's a centromere and they're right next to each other, what sort of centricity is that called? Telocentric. So in this case, chromosome 9 looks like, what do you think? Centromere is not in the middle, so it's not metacentric. So it's submetacentric. What's chromosome 22? What do you think? How would you categorize chromosome 22 shown here? It's either acrocentric or telocentric. It's hard to tell from the drawing. And it turns out in humans, chromosomes 9 and 22 break pretty frequently and exchange parts. And the reason we know this is because when chromosomes 9 and 22 translocate, so they break here in the middle of a gene called ABL, and chromosome 22 breaks right there in the middle of a gene called BCR. And when those chromosomes break and exchange parts, you get one chromosome that's much larger. That's why it's called a plus, 9 plus. And then you get one chromosome that's smaller, and that's termed the Philadelphia chromosome. This causes a type of cancer when this happens. And that's why we know the frequency of this translocation. Because when people get this type of cancer, That's a clue for a cytogeneticist to look at this patient's karyogram, and they say, oh, look, it's the exact same thing that we see happen in almost every other person that has this cancer. It's the Philadelphia chromosome translocation. And it's always chromosomes breaking in the same spots, in the middle of ABL and in the middle of BCR, and the chromosomes switch arms. Which should seem kind of weird. So it suggests there's something about the chromosomes that make them fragile, susceptible to breaking in certain spots, and that's very true of chromosomes. That there are some parts of our chromosomes that are more prone to breaking than others. Chromosomal rearrangements in action. They don't all have to be bad, but sometimes they do end badly, as in cancer.